Assalamu alaikum dear students. Today we are going to study transport, metabolism and excretion of thyroid hormones. As you know, lipid-soluble hormones cannot be stored inside the cells. They are synthesized as required. However, thyroid hormones are an exception. These hormones can be stored in the colloid material. T3 and T4 are bound to thyroglobulin in colloid. When thyroid hormones are released from thyroglobulin and enter into circulation, T4 is abundant, much more than T3. Normally it's 93%. So the ratio between T4 to T3 is about 15 to 1. As these two hormones are highly lipophilic or water insoluble, they cannot be transported in the plasma as such. They need to be bound to some plasma proteins so that they can be easily transported. These plasma proteins are all synthesized from liver, and these proteins are some albumins and globulins. Globulin is a very specific globulin which is called as thyroxine binding globulin, and the other proteins to which these hormones bind, they are called as prealbumin and albumin. The bound form of both of these hormones in our blood plasma are more than 99%, and the unbound or free form is less than 1%. Very high ratio between bound and free form inform us that these hormones are very lipid soluble and have very high affinity for plasma proteins. The free form of T4 hormone is less than 0.1%, and that of free T3 is about 0.5%. This shows us that the affinity of T4 hormone is much more for plasma proteins as compared to T3 hormone. This bound form of T3 and T4 hormones is a significant reservoir of these hormones in our blood plasma, and whenever the free form will decrease, some more bound form will convert into free T3 and T4 hormones. It is the free form of these hormones which performs its biological actions. The fluctuation in the binding proteins, either increase or decrease, can transiently change the bound and free forms of these hormones. For example, in pregnancy, there is increase in estrogen and progesterone hormones. Estrogen especially increases the production of thyroid binding globulin from the liver. This increase in TBG increases the bound form, and the free form can transiently decrease, which has a negative feedback effect on anterior pituitary. So there may be a little bit increase in the production of TSH hormone, which increases the stimulation of thyroid gland. So the thyroid hormone production may increase. So eventually what happens, the total thyroid hormone T3 and T4, they increase, especially the bound form. However, the free form comes back to normal, and clinically this is an euthyroid condition. In case of hepatic failure, decreased production of thyroid binding globulin, that decrease in production of this binding protein will decrease the bound form of the hormone, and the free form can transiently increase, which can have negative feedback effect on the anterior pituitary. Hence, there may be decrease in the production of TSH hormone. That will decrease the stimulation of thyroid gland, and eventually the total T3 and T4 hormone may gradually decrease. However, the free form of hormone will remain normal. These thyroid hormones are released to the tissues slowly. The half-life of T4 hormone is about 6 days, and half-life of T3 hormone is about 1 day. It's because T3 has less affinity to bind with plasma proteins, and T4 has more affinity to bind with plasma proteins. In the peripheral tissues, T4 hormone is converted to T3 hormone. This reaction occurs in most of the tissues of our body, but especially in liver and kidney. Here we need an enzyme which is called as 5' deiodinase or 5' monodeiodinase enzyme. This removes one iodide atom and converts T4 into T3, which is biologically 10 times more active as compared to T4. Another enzyme, 5-deiodinase or 5-monodeiodinase, removes another iodide from another position and converts T4 into reverse T3. Reverse T3 is biologically inactive. It cannot perform its action as compared to T3 hormone. Because T3 is much more active than T4 hormone, that's why some physiologists also call T4 a pro hormone for T3. T3 hormone is more potent and has 10 times more biological activity as compared to T4 hormone. It's because of two reasons. One reason is that it has low affinity to bind with plasma proteins. This is why it has relatively more free form in our plasma as compared to T4 hormone. Secondly, T3 hormone binds very tightly to its receptors in the tissues, and this is why it has more potent effect on its cells. These hormones T3 and T4, when they enter into the target cells, they bind once again to some other proteins where they are stored inside the cells, and then they are used slowly over a period of days to weeks. When injection of a large dose of T4 hormone is given to a person or to an experimental animal, it has been observed that T4 does not start its action quickly. It has a latent period of about 2 to 3 days, after which it starts its action. Then its action gradually increases and reaches to a maximum activity within 10 to 12 days. And this action of T4 hormone persists there for about 6 weeks to 2 months. But when we give an injection of high dose of T3 hormone, the action of T3 hormone starts 4 times quickly. There is less latent period. The action starts within 6 to 12 hours, and the maximum activity of T3 hormone is achieved within 2 to 3 days. The slow start of action of these T3 and T4 hormones is because they are highly lipid soluble hormones and they very tightly bind to plasma proteins, and this is why they are released slowly into the tissues. And they also start their action in the cell very slowly because they affect the genes, and by affecting the transcription and synthesis of new proteins, they have slow onset of action, and their action also persists for longer period of time. Because T4 is having more affinity for plasma proteins, its latent period is much more as compared to T3 hormone, and T3 hormone has a less latent period because it has less affinity for plasma proteins, and also the potency of T3 is much more because T3 hormone binds with its receptors in the tissues very quickly and with more affinity as compared to T4 hormone. Thyroid hormones are metabolized by deiodination, deamination, and also by conjugation reaction. These hormones are conjugated with glucuronic acid. Their conjugated forms are formed in liver, then released in bile into the intestine, and then they are excreted out in feces. Now, I would like to summarize some of the important features of this lipid soluble hormone T3 and T4. I would like to compare the synthesis, storage, transport, metabolism, and excretion of thyroid hormones which are derived from amino acid tyrosine, and compare these features with another derivative of tyrosine amino acid, which is epinephrine and norepinephrine. As you know that epinephrine, norepinephrine, they are water soluble hormones, and T3, T4 hormones, they are lipid soluble hormones. We know that these hormones cannot be stored inside the cells just like other lipid soluble hormones. However, these hormones can be stored outside of the cells in the colloid material. Water soluble hormones, epinephrine and norepinephrine, they can be stored inside the cells in some membrane bound vesicles. These hormones, being very lipid solubles, need some plasma proteins, globulin and albumin, so that they bind with them and are transported in plasma. But epinephrine or norepinephrine, being water soluble, need not to bind with any plasma proteins to be transported in the plasma. As these hormones are very tightly bound to plasma proteins, they have a long half-life and they are released slowly to the tissues. Epinephrine or norepinephrine, they have very small half-life as compared to T3 and T4 hormones. As these hormones are lipid soluble, they can easily cross the cell membrane of their target cells, and they can reach to the nucleus where they can act on the genes and can affect transcription and synthesis of new proteins. This is why their onset of action is slow, because the synthesis of protein is a slow process. But epinephrine or norepinephrine cannot cross the cell membrane of their target cells. Hence, they act on the receptors which are present in the cell membrane and affect the intracellular actions through some second messengers like cyclic AMP, etc. The onset of action of these lipid soluble hormones is slow and the duration of action is long. But for water-soluble hormones like epinephrine, the onset of action is quick and the duration of action is also short-lived. As these hormones are tightly bound to plasma proteins, they are excreted slowly, but epinephrine, those kind of water-soluble hormones, they are excreted rapidly from the body. In our next in our next lecture, we will study the pathophysiology of various diseases of thyroid gland and also the differences between hypo and hyperthyroidism. Thank you so much for watching this video. See you next time with another video.